Thank you very much to the organizers and to everybody here for our opportunity to speak. I'll try not to keep you from your coffee for too long. But we're a San Diego, California-based startup company, and we are developing enabling manufacturing technologies for viral vectors. And I think the take-home message is we have an AAV viral vector farm. So I'd like to walk you through our process and the advantages of producing viral vectors, in particular AAV, using whole plants as modular bioreactors. So I think most of us here are going to be very familiar with the manufacturing problem in the gene therapy vector space, and this is, unfortunately no longer the FDA CBER director said if gene therapy manufacturers cannot streamline manufacturing, gene therapies just will become increasingly inaccessible to the patients that rely on them. And we're seeing this with adoption of these therapies across the globe. So what we have worked on is a completely orthogonal manufacturing system. It's not in cells in culture, it's not in bioreactors, and it has many advantages over existing manufacturing platforms. Our manufacturing solution is highly scalable. It's a linearly scalable model platform for rapid and reproducible scale-out production, and a highly automated manufacturing leveraging already existing industrial agricultural innovations. We have very rapid timelines, particularly for process development. Our modular process reduces manufacturing lead times, and in this case, this is really your process development to get to your locked-in GMP batch by roughly 80%. We can go from your onboarding your gene of interest and your drug product, and in 2 to 3 months, we can get to a complete process development and move forward into clinical scale manufacturing. This process would typically take anything from 8 to 12 months to do process development for a new AAV vector. And we do all of this with a significant cost reduction. We're not talking about a percentage cost reduction here. We're close to an order of magnitude cheaper end-to-end in terms of the process. And we have high yields. Currently, we're producing more than 2E14 VGs per kg of plant biomass. So this is very equivalent to, like, say, a liter of suspension-adapted hexel using conventional triple transfection, with over a 10x reduction in cost of goods and labor costs. And it's very safe and effective. We're growing these in plants, so there's also no adventitious human pathogens in our production platform. We're serotype agnostic. We can produce really any AAV serotype, including engineered capsids. We're gene of interest. Your payload, we're also agnostic to payload. And because your promoter that drives your transgene is going to be a mammalian promoter that is inactive in our plant systems, so we have more reproducibility batch to batch when you're looking at changing out transgene cargo. So just to walk you through the process in a high level, this is a relatively simple process. It's already an industrialized process for producing recombinant proteins and doing gene transfer into tobacco plants. We use nanograms of plasmid as a raw material rather than grams of plasmid that you would transfect into suspension cells. We transform these into a bacteria called Agrobacterium tumefaciens. This is a plant science workhorse and is excellent at transferring DNA into the leaves and the somatic cells of plants. So we're not making transgenic plants. This is a transient gene delivery. It's very flexible and it's very efficient. So we transform plasmid into bacteria. We ferment that bacteria to whatever scale you need for whichever production size. And then we infiltrate this into the leaves of the plants. So this is effectively a pressure infiltration into wild-type plants. This then delivers our packaging constructs into the somatic cells of the leaves. And anything from, say, 3 to 7 days after this process, we're going to harvest the green matter of the plants. We're going to homogenize that and take it through a series of conventional clarification, UFDF, and then into downstream purification using modalities that are very, very common that we use already for mammalian or insect cell-produced AAV, including FPLC-based chromatography methods, ultracentrifugation, and processes that people are very familiar with. So the advantage of this is it's a true scale-out model, as I suggest, is we have unparalleled efficiency, we have exceptional plasmid utilization, and very aggressive cost of goods. What we're building in San Diego over the next few months is a fully automated pilot production facility. There's also going to dramatically reduce labor costs, and we have flexible manufacturing and a very rapid process development turnaround time. So as a comparison to the process for process development using conventional bioreactor systems, as I sort of mentioned, your process development for an AAV vector that you onboard, either you're doing it yourself at GMP production or you're working with a CDMO, can take anything from like 8 to 12 months. Because this is a scale-up process. You're going to start in very small bioreactors, you're going to step up in size until you get to your clinical production volume, and each step requires reoptimization. You also have to maintain a seed train of your cells going into the bioreactor, and seeding into a bioreactor for a production run can take 1 to 2 months to amplify those cells. So overall, the process takes 8 to 12 months, and to just get to your first locked-in GMP batch is going to cost you at least $1 million, depending on production size. We can do the same process in about 2 to 3 months, because the way that the vector is produced in each individual plant is stereotyped. We can do rapid iterations of process development at small scale, say 10 to 20 plants. That's in terms of yield equivalent to about 1 liter to 2 liters of suspension-adapted HEC cells. And then once we have quality and yield data, we can immediately go to commercial scale. So this process to get to your locked-in GMP batch can take 2 to 3 months and at a significant cost reduction. So we're very lucky at the end of last year to being competitive to receive an ARPA-H contract award of up to $61 million that is taking our research scale production and allowing us to build a pilot production facility. This will allow us to establish full cGMP operations, expand the platform's impact to additional viral vectors that we're starting to do feasibility and proof of concept studies with, and also take a lot of our AVs that we can produce in-house into additional in vivo comparability studies. So we're building a 35,000 square foot US-based process developed manufacturing facility that we acquired at the beginning of this year. The pilot scale capacity is going to be 1E16 VGs per week, and we're looking at initiating and commissioning that by Q3 2025. And then final commercial scale capacity is approximately 1E17 VGs per week, and we're looking at the second half of 2026 for that facility. And what you're looking at really is a schematic of the vertical farm systems that we're using as our upstream horsepower. This whole process is automated and segregated, and the beauty of it is you can manufacture a GMP product where your upstream environment is in controlled non-classified space rather than full clean room space, and that's acceptable from a regulatory perspective for a clinical product. So we're really driving transformation in gene therapy through partnerships, and we're very interested in co-development programs with early phase drug developers to do feasibility and to work with them through into IND. So I want to very quickly go through some of the data, and then if we have time at the end, address any questions. So we've manufactured plant-produced AAV, and then we manufactured the same AV9 GFP vector in HEC cells. And really what I'm going to show you is we have very comparable quality of vector by analytics. This is proven safety against industry standard tests, and also superior commercial products, both in terms of speed and cost. But we took these vectors into NHP experiments to do a large animal and a clinically relevant species, looking at systemic delivery of these two vectors. So first, the QC release testing of the vectors that are produced in plants compared to mammalian. We have, in the crude lysate, we have very good empty-full ratios. After in the purified product, so these were purified by affinity, capture, FPLC chromatography, and then enriched by ultracentrifugation. In the drug product, we have very high full ratios in both the plant and the mammalian. They're very comparable. The drug product that we dosed into the primates were titer matched, and we confirmed the titer both by qPCR and dPCR, and we also did that externally with the CRO to validate it. Because we have a bacteria that we use in our delivery process, we obviously needed to ensure that we have low or no endotoxin, and in both preps we have endotoxin levels that are way less than the FDA standards. And our sterility, our bacteriostasis, as well as mycoplasma, we've also tested for spiroplasma, and we're developing analytical assays for plant-specific residuals, are all within spec, and these vectors are very comparable in terms of their biophysical characteristics and their quality release characteristics. We've also looked at this with orthogonal methods, including Stunner. We've done mass spec to look at PTMs. I'm happy to talk about that to anybody after this, and also electron microscopy to characterize the particles. So I won't go through the study design. We did in too much detail, but we dosed this systemically into groups of primates. We also dosed it into the CSF intravitreally, and then it was a 6-week in-life study, and we really, as a sort of a non-GLP farm tox study. I think here what we're looking at is the expression of our transgene in the plant-treated group compared, normalized to the HEK-cell-treated group data. So we saw very similar biodistribution, but from the same titer of vector that we administered, we see higher transgene expression in a number of the target tissues that are you'd expect for AV9, including skeletal muscle, sometimes up to 13-fold higher transgene expression. So we're actually very interested into the mechanism of this. We know it's not driven by PTMs. We're doing some genome characterization of the AAV right now to see if we sort of understand that mechanism. This is just showing, we also looked at the protein expression. We get very robust protein expression as well. This is in lumbar spinal cord. If we're looking at markers for inflammation, both in the liver and in the CNS, looking at ALT as well as GFAP expression in the CNS, we see really no change in any of these markers of inflammation between the two treatment groups and between the control groups. So our vectors, both the plant and the HEK-cell vectors that we generated, are very well tolerated. But the really interesting result from the primate data is we looked at the in-life portion as well as at the endpoint of the study, is in the animals that were dosed with the plant-produced AAV, we always see much lower neutralizing antibody responses. And again, this is for systemically administered AAV9. And this really opens up the possibility of redosing using interventions that, to reduce IgG when you have a much, much lower NAB response from the outset. So we're very interested in pursuing this, seeing if it's true across all of the serotypes and payloads, and understanding what the mechanism of this is. So just to sort of wrap it up, to say we believe that this is truly a paradigm-shifting technology that is going to enable production of AAV to meet the global demands and actually allow for equitable access of this for patients. It's reliable, it's safe, we have very good efficiency, there's excellent comparability and compatibility, and we have the potential for much more facile redosing with a much lower immunogenic profile and much lower neutralizing antibody responses. So with that, I'd just like to wrap it up. We're funded by Insight Partners, KDT, and Five Fund. I'm Dan Gibbs. I'm one of the founders and the CEO, and also we have the other founder, Jake Connors, and the CTO here, and our head of BD is sitting in the audience, Adam Roos. If you're interested in partnering with us, please come talk to any of us here. And we have an excellent scientific team, and I'll leave it there. Please come and meet us at the meeting and see if you want to grow some AAV. Thank you very much.
Thank you.